Hi, my group is group 17. My group have 3 people. First, me, not Shawani Fitriah. Next, Sharatu P. Ramzani. Lastly, Suraya. My group can present about cell division for mitosis and meiosis.
Mitosis. Mitosis is a part of the cell cycle in which replicated chromosomes are separated new into two new nuclei. Next, the process of division of eukaryotic cell nuclei to produce two offspring cell containing the chromosomes that are identical to the parent chromosome, have the same number and type of chromosome as the stem cell. Mitosis is involved in the process of growth, regeneration and replacement of damaged or all cells. Mitosis occurs in all somal cells. Example, the body cell of the organism other than gamete cell, reproductive cells such as sperma and ovum. Mitosis occurs in plant cell and animal cell in specialized tissues. The second, the process of mitosis. In prophase, the chromatin in the nucleus condenses into chromosome. The membrane around the nucleus breaks down, centriole, divides, and move to pole. Formation of ester formation of spindle fibers. Next, in metaphase, the chromosome line up in the middle of the cell centromeres attached to spindle fiber. In anaphase, each chromosome breaks up, centromere split into two chromatids. Chromatids are pulled to opposite poles of the cell formation of cleavage furrow, beginning of Cytokinesis. In Telophase, mitotic spindle disappear chromosome unwind to chromatin. New nuclear membrane form around each group of chromatin after telophase cytokinesis completed. After mitosis, cytokinesis occurs to form two new cells, and the two new cells start the cell cycle again. Besides the important information of mitosis, first, a daughter cell have the same chromosome number as to the parent. Second, daughter cells are genetically identical and chromatic crossing over not occur. Third, cytokinesis occur once. Four, occurs in somatic cell. 5, product the diploid cell and 2 cells. 6, is occur to a circle. The mitosis have two species. First, mitosis in plant, and second, mitosis in animal. Okay, I want explain the mitosis in animal. First, unlike plants, growth in animals is not limited to specific body parts but occur in almost all body parts of animals. The cells that are in the Malpighi layer, the deepest layer of epidermal tissue of skin, undergo mitosis to form new cells. These new cells will replace the injured cell and the dead cell on the outer surface of the keratinized outer skin. On the Malpighi layer, there is also a melanin pigment that gives a color to the skin. Nerve cells will stop carrying mitosis as soon as a person reaches a certain age in his life. Then mitosis in plant. In plant, growth occur only in certain part, namely at the end of the shoot and roots and in the cambium. These parts have meristem cells that actively divide by mitosis. The meristem cells found at the root and end of the shoots are known as apart meristem cells. Apart meristem cells are responsible for plant growth. The meristem cells are found on the lateral edge of the decanal trunk as the lateral meristem or cambium. Side meristem or cambium is important for increasing the size or plant the stem circumference. Lastly, the reason why mitosis. First, to allow multicellular organisms to grow and develop. Second, to replace and repair other cells that have been damaged or worn out. Third, for asexual reproduction, example binary fission. For regeneration, some organisms can regenerate body parts. The production of new cells in such instances is achieved by mitosis. For example, starfish regenerate lost arms through mitosis.
Meiosis, Prophase One. In this stage, the DNA condenses to form chromosome. The duplicated sister chromatids join together at the centromere. They stay fused at the centromere till Meiosis One. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving two pairs of sister chromatids. Chromosome material is exchanged between the two pairs of sister chromatids. This event is called recombination or more commonly crossing over. This is one of the reasons why the two siblings, aside from the twins, are genetically identical. There are more key steps in Prophase One. The nuclear membrane starts to break down. Then the two centromeres migrate to opposite ends of the cell and microtubules appear. The microtubules then attach at the chromosome. Metaphase One. The synapsis chromosome align at the equator of the cell. The chromosome align randomly, which result in different combination each time meiosis occur. Anaphase One. During this phase, homologous chromosomes separate and migrate to the two poles of the cell. The sister chromatids remain attached at the centromere. Telophase One and Cytokinesis. The cells are divided into two daughter cells. Each of these two cells undergo meiosis II. Prophase Two. Again, chromosome condense. The nuclear envelope breaks down and the spindle apparatus form. The differences between Prophase One and Prophase Two are the daughter cell only have one copy of each homologous chromosome. So, in the Prophase Two, there is no synapsis homologous chromosome or crossing over. Metaphase Two. The chromosome align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for this chromosome to align. Anaphase 2. The sister chromatids are pulled apart as the microtubules shorten. Also, the end of the cells are pushed further apart as microtubules elongated. Telophase 2 and cytokinesis. The nuclear membrane reforms. and the cytoplasm is divided into two haploid daughter cells. The division is called cytokinesis. Since meiosis begins with two cells and each of the cells was split into two cells, four unique haploid cells form at the end of meiosis. The Meiosis Mistake. This is a picture of trisomy children. One of the original cells had an extra chromosome. People with trisomies have 3 copies of a particular chromosome instead of 2. This means these individuals have a total of 47 chromosomes and plus 1. A fairly common and aneuploidy that involves an extra chromosome 21. This is also called Down syndrome. It affects about in every 750 babies born in Canada. Children with trisomy 21 may experience delay when learning to crawl, walk, and speak. As they get older, they may have trouble with reasoning and understanding. Triple X syndrome. It is associated with learning disabilities and organ abnormalities. Klinefelter syndrome. In a male fetus, the result of an extra X chromosome XXY. Males with this condition have smaller testicles and are infertile. Jacob syndrome. Males born with an extra Y chromosome XXY. The symptoms include language difficulties, problem with sitting and walking, and behavior emotional issues. People who have an extra Y chromosome may also have mild autism and weak muscle tone, hypotonical.
Thank you for watching our video. Before I end the video, I remember for you all to stay safe and stay healthy. Thank you.